Imagine a world where extinctions aren't the end of the story. A world where creatures that vanished thousands of years ago could be brought back to life, walking the earth once more, roaming their ancient habitats. This isn't mere science fiction. It's a burgeoning and controversial scientific field known as de-extinction, which stands on the shoulders of remarkable advancements in the study of ancient DNA, aDNA. Understanding how ancient DNA works and the techniques that allow us to extract and analyze it opens the door to profound existential questions about the ethics of science, our role as humans in preserving biodiversity, and the limits of our ability to manipulate nature. In this article, we'll dive deep into this astonishing topic, exploring how scientists are recreating extinct creatures, with a particular focus on the Woolly Mammoth project as a pioneering and discussion-provoking example. A journey to the past, understanding ancient DNA. Before we discuss bringing extinct creatures back, we must understand the essence of the tool that makes it possible: ancient DNA. DNA is the genetic blueprint of life, carrying all the instructions needed to build and operate an organism. When an organism dies, its DNA gradually begins to degrade. However, under specific conditions, such as very low temperatures, permafrost, extremely dry conditions, deserts, or protected dark caves, DNA can remain intact for thousands, even millions of years. The study of ancient DNA is a relatively new field emerging in the 1980s, and it has undergone a true revolution thanks to developments in genetic sequencing technologies. Initially, extracting and amplifying ancient DNA was a laborious and limited task, with contaminated samples posing a significant challenge. But with the advent of techniques like polymerase chain reaction, or PCR, which allows for the amplification of very small amounts of DNA, it became possible to study samples previously inaccessible. Unique challenges of ancient DNA. Limited quantity. The amount of available DNA in ancient samples is typically minuscule. Fragmentation and degradation. Ancient DNA is subject to damage and fragmentation over time, making it difficult to read. Contamination. Ancient samples are often contaminated with DNA from bacteria, fungi, or even human DNA from handlers. Biological complexity. Understanding the biological function of ancient genes requires advanced analysis and comparisons with living species. Despite these challenges, scientists have successfully extracted DNA from mammoth bones, teeth, hair, and even from plant and animal remains preserved in ice or caves. This DNA has not only revealed evolutionary relationships between extinct and living species, but has also provided unprecedented insights into the genomes of these creatures, shedding light on their physical traits, behaviors, and even the causes of their extinction.
De-extinction. Can life be turned back? The term de-extinction simply means bringing extinct species back to life. There are several approaches to achieving this, but the most common and applicable relies on ancient DNA and genetic engineering techniques. Cloning. This is theoretically the most direct method, where a viable cell is taken from an extinct organism, its nucleus is extracted, and then it's implanted into an enucleated egg cell from a closely related living species. The egg is then stimulated to grow and develop into an embryo, which is implanted into a surrogate mother. The main problem here is the scarcity of complete and viable cells from extinct organisms, in addition to the challenges associated with finding suitable surrogate mothers. Genetic engineering. This approach is currently the most realistic and is used in the Woolly Mammoth project. Instead of cloning the entire organism, scientists analyze the DNA of the extinct creature and compare it to the DNA of its closest living relatives. Then, gene editing techniques like CRISPR-Cas9 are used to modify the DNA of the living species to introduce the genetic traits of the extinct species. The idea is not an exact clone, but rather creating a hybrid that possesses most of the distinctive characteristics of the extinct species. Selective breeding. This method does not directly rely on ancient DNA, but rather on breeding living species with traits similar to extinct ones with the aim of re-establishing those traits. An example is the Taurus project, which aims to recreate the aurochs by breeding domestic cattle breeds with similar characteristics. The Woolly Mammoth project, an icon of de-extinction. The project to revive the woolly mammoth is the most prominent and ambitious example in the field of de-extinction. Woolly mammoths were massive elephants that lived during the Ice Age and went extinct around 4,000 years ago. Their closest living relatives today, Asian elephants, live in warm and entirely different environments than the mammoth. Why the woolly mammoth? Availability of DNA. A large number of well-preserved mammoth remains have been found in the permafrost of Siberia, providing a rich source of ancient DNA. Distinct genetic traits. Woolly mammoths possessed clear distinguishing features from modern elephants, such as thick fur to protect them from the cold, a thick layer of fat, small ears to minimize heat loss, and massive tusks. These traits are encoded in their genome. Potential ecological role. Some scientists believe that the return of mammoths could help restore the mammoth steppe, a vast grassland ecosystem that covered large parts of Eurasia and North America during the Ice Age. It's thought that heavy mammoth grazing maintained these grasslands from turning into dense forests, allowing for more carbon sequestration and reducing methane emissions from thawing permafrost.
Leading companies and initiatives. Several research teams and companies are working on the Woolly Mammoth project, most notably Colossal Biosciences, co-founded by renowned geneticist George Church from Harvard University. Colossal aims to bring back the woolly mammoth not as a complete clone, but as a cold-resistant elephant hybrid possessing most of the key mammoth traits. How is it done? Steps and challenges. Mammoth DNA sequencing. The full genome of the woolly mammoth has been sequenced multiple times, allowing scientists to identify genes responsible for its distinctive traits like thick fur and fat. Genome comparison. The mammoth's genome is compared to its closest relative, the Asian elephant, to identify key differences. Gene editing. Using CRISPR-Cas9 technology, Asian elephant stem cells are modified to introduce the distinct mammoth genes. For example, genes for proteins responsible for thick hair growth or those controlling fat tissue production can be modified. Embryo implantation. After cell modification, they are converted into embryos and implanted into Asian elephant surrogate mothers. This is the most challenging step in the process, as elephant gestation takes about 22 months and many surrogate mothers are needed. Colossal is working to develop an artificial womb to simplify this process and reduce the burden on mother elephants. Ecological reintroduction. If successful, the goal is to reintroduce these hybrid mammoths into areas of Siberian permafrost, such as Pleistocene Park, a Russian initiative aimed at restoring the Ice Age ecosystem by reintroducing large species like bison, reindeer, and horses. Ethical and environmental implications of de-extinction. While the de-extinction project promises a stunning future, it also raises a number of complex ethical and environmental questions that must be seriously addressed. Ethical considerations. Animal welfare. Is it ethical to subject Asian elephants to lengthy surrogate pregnancies and births, especially if these hybrid embryos may not be fully viable? And what quality of life would a recreated creature experience in an environment that perhaps no longer exists in the form it once knew? Human responsibility. Do we have the right to manipulate life in this way? And what are the limits of our intervention in natural evolutionary processes? Costs and benefits. Could the enormous resources invested in de-extinction be better used in conservation efforts for currently endangered living species? Environmental considerations. Changing environment. Will the recreated creatures be able to adapt to today's environments, which have significantly changed since their extinction? For example, the global climate today is warmer than it was during the Ice Age. Diseases. Could recreated creatures carry new or unknown diseases that could spread to living species? Ecosystem impact. What is the impact of introducing extinct creatures on current ecosystems? Will they compete with living species for resources? Will they disrupt food chains? Shifting baseline syndrome. Some critics fear that the idea of de-extinction might lessen the urgency of conserving current living species, creating an illusion that we can always fix the damage we inflict on nature later. Beyond the mammoth. Other examples and the future of de-extinction. The ambition of de-extinction is not limited to the woolly mammoth. Several other projects are exploring the possibility of bringing other species back to life, including dodo bird, a large, flightless bird that lived on the island of Mauritius and went extinct in the 17th century due to human activity. Its closest living relative is the Nicobar pigeon. Thylacine, Tasmanian tiger, a carnivorous marsupial that went extinct in the early 20th century, with living relatives in Australia. Pyrenean ibex, a subspecies of mountain goat that went extinct in 2000. In 2003, a calf of this species was cloned, but it died minutes later due to birth defects. This was the first time an extinct creature was brought back to life, even if only for a very brief period. Passenger pigeon, a bird that was once the most numerous in North America and went extinct in the early 20th century due to overhunting. The potential future of de-extinction. Advances in DNA sequencing and gene editing technologies are accelerating at an incredible pace. Within a few decades, we might see a generation of hybrid creatures bearing the traits of extinct species. However, the true success of de-extinction will not only be in creating these creatures, but in our ability to return them to environments where they can thrive without harming existing ecosystems. Perhaps the ultimate goal isn't to bring back every extinct species, but rather to gain a deeper understanding of biology, improve conservation tools for living species, and restore some lost ecological functions. This research might help us develop disease resistance in living species or understand how organisms adapt to climate change. Between promise and precaution. The project of bringing back extinct creatures, exemplified by the Woolly Mammoth project, represents a qualitative leap in our understanding of biology and our ability to manipulate life. It is a project that holds great promise for restoring some of the biodiversity we have lost and for sparking vital discussions about the future of environmental conservation. However, this promise comes with serious caveats. Scientists, policymakers, and the public must approach these developments with extreme caution, taking into account the long-term ethical and environmental implications. Should we do everything that is technologically possible? Can we bear the consequences of releasing extinct creatures into a changed world? Ultimately, the goal of de-extinction may not be to fix the past, but rather to serve as a powerful reminder of our collective responsibility to preserve life on our planet, and to ensure that we learn from past mistakes instead of undoing them. It's a lesson in scientific humility and a call to action for a better future for all species, extinct and extant alike.